installment of the Geology lecture series at Southwestern Oregon Community College, as we have gone virtual, at least temporarily. So it's actually a year ago today, because this is being archived, and I thought it would be intriguing. Today, I noticed on the news that this was the anniversary of the first case of COVID in Coos County. I also noticed on the OHA report that Coos County had 0 cases reported today, which is fantastic. Those of you local probably already know that we've transitioned from extreme to high, so hopefully this is a trend that will continue. Just looking ahead, our final talks in the lecture series this year are also going to be via live stream. They're scheduled for May 11th, a little bit earlier, 3 p.m. and 4:15 p.m. We've got a double header of 2 speakers that are IRIS SSA distinguished lecturers. The first one at 3 p.m., Dr. Guojing Ling from University of Miami is going to talk about the 2018 Kilauea volcano eruption, expected or a surprise, and a link to the seismic activity associated with that. And the other speaker is Dr. Ben Holtzman from Lamont Doherty, a part of Columbia University, on seismology with your ears, listening to patterns in tectonic, volcanic, and human-induced earthquakes. So that double header will be exactly 51 weeks after our first go-round with virtual talks. And hopefully that will be a precursor to us going live in the Hale Center next fall, in the fall of 2021, and through the remainder of that academic year. I'd like to take just a second to thank our sponsors that you saw earlier: DB Western, The Mill, Southwestern Foundation, IRIS SSA, the college, and also the the Ocean Discovery Lecture Series, which brings tonight's speaker, Dr. Jessica Labonté from Texas A&M University. Dr. Labonté went to Laval University, where she grew up in Quebec City, Quebec. Then she went and worked on her Ph.D. at the University of British Columbia, and currently is at Texas A&M University. And so without further ado, it's my pleasure to introduce to you this evening Dr. Jessica Labonté with You Are What You Eat. Thank you.
Hi everyone. First of all, I would like to thank you for inviting me to give a presentation to you today. This is an IODP, so International Ocean Discovery Lecture. So I'm going to tell you a little bit, on top of my research, I'll also tell you a little bit about the ocean drilling program. But my presentation is entitled You Are What You Eat, a geochemical and microbial study of a 3,000-year-old stratigraphic sediment succession. So we're going to talk about sediment, we're going to talk about chemistry and microbes. So when I think about the world, I think of a world that is microbial. And when we're thinking about subsurface environments, that's especially true. So the microbial biomass on Earth, it's about 77 gigatons. And when I talk about microbes, I think about archaea, viruses, and bacteria. There's also protists, but for our studies, we are focusing on archaea, viruses, and bacteria. And there's about, of these 77 gigatons of carbon, there's about 70 gigatons of carbon that is only in the deep subsurface. And when we look at the subsurface, which is here in black, it is totally dominated by bacteria and archaea. We don't find plants, fungi, protists, and animal. If we find these, it's because they have been buried. And because they're so abundant, they're also playing important roles. And when I think about subsurface, what I mean is the deep continental subsurface. There can be soil, but it's mainly continental subsurface, oceanic sediment, and deep oceanic subsurface, like below the sediment. But today, in the study that I'm going to tell you about, it's coastal sediment. There's also a lot of microbes in the water column, but when you look in terms of where the microbes are, the majority of them are found in the subsurface. And they play really important roles. So in the water column, there's a lot of things that are happening, from primary production to respiration. But when we're thinking what are sediment, it really is like all these particles that are sinking at the bottom of the seafloor, and they contain organic matter, microbes, and they are fueling microbial decomposition. So there's a lot of respiration, methanogenesis, remineralization that is happening in the subsurface. But there's also production of fossil fuels. So they are super important to maintain our planet, and they're important in the balance of the geochemical cycles. Carbon, very important, but also sulfur and nitrogen. And when we think about sediment, why are they important? Well, they cover more than 70% of our planet. And there's different depth, but in areas that are not very active, like the subtropical gyre, or there's not a lot of the thickness of the sediment is quite thin. But when we go in environments that have a really high growth rate and turnover rate, then sediment thickness can be up to 10 kilometers. 10 kilometers, that's 6 miles deep of sediment. And microbes can live quite far deep down these sediments. Actually, there was one study where, because it's hard to access these microbes, but there's one study where they were able to get sediment cores up to 2.5 kilometers. That's almost 3 miles below the seafloor. And they were still finding microbes that deep. And you can see here, so here we have a histogram where each bar is one sample, and you can see the distribution of microbes through each of these samples. So in the shallow part, up to about 300 meters below seafloor, there's this group there's this community that is living, and then below there's a different kind of community that is living where it's deep. But the important point is that they did find microbes 2.5 kilometers below the seafloor. So we see here that there's some changes in each of the samples. The microbial communities, they fluctuate. What are the drivers of these microbial communities? These are some of the questions that I'm trying to answer in my lab. So one of the things that we know is nutrient availability. So when you're thinking about nutrients, how do microbes make their energy? They make their energy from these oxidation reduction reactions, which will and these will generate energy to be able to make ATP, which is the energy currency of the cell. And here we have the redox potential for some common reactions. So basically, these reactions here, they are presented as a tower, and you have the higher the drop, the more energy will be released by the reaction. So oxygen is usually a good electron acceptor. So you have your electron donors, electron acceptors, and the bigger the drop, the more energy you're making. And the more energy you're making, you're going to be able to produce more ATP. So, but in the sediment, especially in the system we're studying, oxygen isn't really an option. It is anoxic. So you need to go and find other compounds that will be part of these redox reaction to be able to make energy. So how is this nutrient availability playing into these systems? So in the deep sea, in places where there's a slow sedimentation rate, so places where there was a not very thick sediment, usually you're gonna have oxic sediment, but because there's not a lot of sedimentation, there's not a lot of carbon coming. And carbon is a really good electron donor because the organic matter is electron donor and it's full of carbon. But here, you don't have a lot of carbon. So microbes are, there's a lot of electron acceptor, but not a lot of electron donors. Where the sedimentation rate is high, and that is the case for coastal sediment, then you're going to have anoxic environment, but also carbon rich. So you have a lot of electron donors, then you get limited by your electron acceptors. Another driver of microbial activity is microbial diversity, is viral activity. And here we have organic rich mud sediment from the Baltic Sea. And what we see in red, we have the viruses. In black, we have the total cell counts. And in green, the green triangles, we have total organic matter. And we see that the viral abundance increases with depth below the seafloor. And then it will peak at the same place where the total organic carbon is peaking, and then it will decline with depth. And that's a really interesting trend, that it peaks with total organic carbon. Another study has looked at different places on the planet, whether it's places that are very slow microbial activity because there's not a lot of organic matter, like the South Pacific gyre or coastal sediment, and they measure the virus to cell ratio. So in marine systems, the virus to cell ratio is usually about 10. You have 10 more viruses than you have cells. And they found interesting trends where in the sediment, the virus to cell ratio is actually much higher. So we're not quite sure what this means. Is it that there's more viruses that are produced, or is it simply that viruses can be preserved for a longer period of time? By looking at viral activity, we could answer these questions, but right now, based on counts, we cannot really say exactly what's going on. But we know that viruses, they play very important roles, ecological roles in subsurface environments. So here I'm just going to refresh your memory on what the typical virus cycle is. So I'm talking about viruses in general. This is a general term because it encompasses all the viruses, but for viruses infecting bacteria, it would be a bacteriophage or phage. Viruses infecting archaea or archaeal viruses. So because in our system we have both, I will use the term viruses the whole time. But here I'm going to use phage because this is more common for phages. When a phage will encounter a host, it usually just finds its host, attach, sends its DNA through the membrane, and then it will hijack the cell completely to make more viruses, and it will end in the lysis or the burst of the cell, which will release its inside, which are nutrients. But some viruses, some phages, they have the ability to actually integrate their genome inside the one of their hosts. So they're not killing their host, they're going in there in the genome, and they're dormant, and they will replicate at the same speed as their host. In that case, they will be called a prophage. And then if conditions arise, they may be able to go back into the lytic cycle. So that has an impact which way the viruses are going on on the environment itself. So in sediment that has in environments that have a low sedimentation rate, well, there's not a lot of carbon, not a lot of cells, so the encounter rate, trying to find a virus in a place where there's not a lot of viruses, not a lot of cells, is not really easy. So in that case, it may be more advantageous for a phage to be integrated inside of its host as a prophage. But in environments that are carbon rich, where there's a lot of microbial growth, there will be lots of cells, lots of viruses. The encounter rate is bigger, so then it is higher. So you may be it's maybe more advantageous for the viruses to go through the lytic cycle because then every time a cell bursts, it releases its insides, its food for the neighboring microbes. So I've told you a little bit about microbes in the subsurface. How do we access the subsurface, especially in the ocean? And that's one of the big mission of the International Ocean Discovery Program, which I'm going to present to you right now. So scientific ocean drilling, it started in 1958 to 1966, and at the time it was called Project Mohole, and that was to reach the barrier, the limit between the mantle and the earth crust, which is called the So far I've told you about microbes that live in the subsurface, but you may be interested in learning how we can access the subsurface, especially when we're thinking about the ocean. And that's really the mission of the International Ocean Discovery Program. So I'm going to tell you a little bit about the history of the program. Scientific ocean drilling, it started with Project Mohole in 1958-1966. That was to reach the barrier between the mantle and the earth crust, and to see how deep we can drill, basically, and how the earth is actually made. But this is still not completed, because it's very difficult, because once you get a place that is too hot, it will either seize in the drill bit, or it's so deep that we don't have drills that are good enough to actually reach the mantle. But after this Project Mohole, it became the deep sea drilling program, which was US funded for about 5 years, and then it became international, and up to this day it is still an international program. In 1985 to 2003, it was called the Ocean Drilling Program, and 2003 to 2013 was the Integrated Ocean Drilling Program, that first edition of the IODP, and then from 2013 to 2023, it was the second edition of IODP, International Ocean Discovery Program, and since 2013, microbiologists are on board constantly. Almost every expedition has its microbiologist. There are multiple platforms associated with IODP. The JOIDES resolution is operated by the US. We can see it here or in the background of my slides. It's a riserless platform. It's sort of the jack of all trade of drilling vessels. It can go in many places, most places on Earth. The CHIQ is operated by Japan. It has a riser platform, and it allows it to drill very, very deep. That's how they got to 2.5 kilometers below the seafloor. There's also mission-specific platforms that are operated by a consortium of European countries, and these are platforms that are chosen based on the specific needs and objectives. So although the JOIDES resolution is sort of a jack-of-all-trade ships, it cannot drill everywhere. So for example, when they drilled the Atlantis Massif, which is this giant rock in the middle of the Atlantic, it was too hard to drill for the JOIDES resolution, so they used a mission-specific platform. They actually built these robots that went down at the bottom of the seafloor and drilled in the rock of the Atlantis Massif. If you are interested in participating in IODP activities, you can sail as a shipboard scientist or technician. You can serve on an IODP Science Advisory Panel. You can serve on the US Science Advisory Committee. My term ended last year, but I was a member of the USAC committee for 3 years. You can also request your own sample to work on them on your own research. And there's also graduate student opportunities. You can sail. They have the Schlanger Ocean Drilling Fellowship for graduate student. And you can also assist in post-course research. And if you want to learn more about IODP, you can go at usoceandiscovery.org. So now I'm going to go back to talk a little bit about my research. So unfortunately, I wanted to talk about one of my IODP project, but it wasn't possible because of COVID. We were not able to get the data on time. So I'm going to tell you about another project that that was done in coastal sediment in the Bahamas. So the goal of this study was what are the drivers of the microbial communities in coastal anoxic sediments. So that stemmed from an IODP expedition 327, Juan de Fuca Ridge Flank Hydrogeology. So they went and they sampled cores to see if there was a difference between the microbial communities between the outcrop as you get away from the outcrop. So how does the seafloor work? You have water that will come in, and this water will interact with the rock and the fluids and chemically change and until it goes out in the discharge. But there's also exchange between the flowing water and that's in the basalt and the sediment. So the idea was, well, as the water chemistry of the water is changing, the microbes should also be changing as we move away from the outcrop or closer to potentially where the water is going in. So here we have the meter, like the depth of our samples. We have sediments that are the 6 at the bottom and basalt, and they go from closer to the outcrop with red, blue to further away. And what we noticed is that there wasn't much difference whether you were like closer or further away from the outcrop, but the differences, so the basalt were obviously different, but basalt, it's rock, and trying to get cells off of rock is not always very easy. But when we looked at the sediment, there isn't much difference, but the differences, although they were small, were mainly based on the composition of the sediment. So if you had a lot of clays, like you could see the nitrospira, versus if you have the forams. So it was based on texture or the kind of sediment more than the geochemistry of the water. So then I was curious about this, and I'm like, if we have a core where we can actually sample the different textures, are we going to be able to say anything about the microbial communities and how they're changing? So we sampled, our study site was Blackwood Sinkhole, which is located in the Bahamas. It is, so the water column is anoxic as well as the sediment. It has a fast sedimentation rate. So if you remember, fast sedimentation rate means you probably higher microbial activity, and although it's organic rich, there may be a potential lack in terms of electron acceptor. So it's rich in total organic carbon, 14 to 29% of the total weight, and what's interesting is that it's groundwater fed. So this is fresh water, but it's very close to the beach, 220 meters away from the beach here, and because sinkholes, they are like the rock is dissolving and it reached salt water. So the bottom is salt water fed, the top is fresh water. So we had 3 questions. Are the subseafloor prokaryotic communities comparable to the ones inhabiting anoxic oceanic basins? What drives the microbial community composition in anoxic coastal sediment? And what is the extent of nutrient regeneration in subseafloor sediment, in the nutrient regeneration by viruses? So we collected a fresh core in July 2018, and so this is our core, and we separated it in 24 stratigraphic layers based on the color and the texture. And for each layer, we did total DNA extraction for 16S ribosomal RNA gene amplification. We did the C to N ratio. And for 6 of them, we did viral production experiments. We did total DNA extraction for metagenomic sequencing. We couldn't do that for all of the samples because it requires a lot of sample, up to 50 grams, to get enough DNA viruses. Although they are very abundant, they're also very small. So we need to get more to be able to get enough DNA for sequencing. We also did radiocarbon dating on 5 samples, and we did pore water geochemistry on the same samples that we did metagenomic sequencing. And our core was almost to the center, and so it's that dark star here. But there's other studies that have been done at Blackwood Sinkhole, and they were done on other cores, whether they were in the periphery or closer to our core. So the first question was, how old is the core and how does it compare to other cores from Blackwood Sinkhole? So to do that, we did radiocarbon dating and measurement of total carbon and nitrogen and measured the C/N ratio. So how old is the core? We had our radiocarbon dating samples, and it's about almost 2,000 years old. We went almost to 2,000 years. And then we did the C/N ratio, and we can see that it fluctuates overall. It is not at a super fine scale, but it is enough to be able to compare our microbial communities to the C/N ratio. And there's some that it's very high, some that it's lower. How does it compare to other cores? So in the periphery, they were able to go to almost 3,000, but our core is short. It's about 90 centimeters, which is about the age that it would be on other cores. And to do that, so what my collaborator, Piet van Hengstum, he's a micropaleontologist, and what he does, he reconstructs the climate using radiocarbon dating to date, and he's looking, and he's also looking at the grain size, whether it's fine or coarse, and the C/N ratio. If there's a lot of leaves, if it's high, it's usually because there's a lot of leaves, and low when there's more algae coming from the sedimentation from inside the core. And he reconstructs the climate, because if you have an event like a hurricane, well, you're gonna get a lot of coarse grain and high C/N ratio. So he's able to look. So here we had Hurricane Floyd, Jean, and then but there are some places that there were probably pretty heavy storms, and he's able to look at that through time. Another study that has been done at Blackwood Sinkhole is that they looked at the source of organic matter. So by looking at the C/N ratio, you can compare it to other environments that are similar, whether it's terrestrial organic matter, because the sinkhole is terrestrial itself. So there may be like organic matter coming from the terrestrial environment, or wetland, which is even more similar to what the environment is, or coastal. And it looks like so our core is the dark blue, is heavily influenced by wetlands, which is a mix of terrestrial and water. Another study that has been done, and this one is important in a sense that our data fit this study the most. They looked at the Blackwood Sinkhole's vegetation history over the past 3,000 years. So they looked at leaf products to determine what kind of trees were surrounding, and also what kind of algae were in the sinkhole. And you had your tropical hardwoods that were in the early days, and then you have it shifted to a pine. There was a lot of mangroves also at one point. So you see this shift of vegetation, and I'm going to talk a little bit about this throughout the presentation. And what this study did is it also divided the time of the sinkhole into 3 age groups or 3 periods. So the most recent is a mix of marine and terrestrial, mangrove origin, or like, so where is the organic matter coming from in general, and based on the vegetation. So marine and terrestrial mangroves is the most recent. Then there was this elevated aquatic productivity, which is so here through time you have your most recent mangrove, then you have your productivity, and that's a very short period of time, and then you have your terrestrial derived vegetation, which is the oldest part of the core. So I already mentioned it, but this is something I'm going to come again throughout the presentation. But if you have a high C/N ratio, means that you have more leaves and cellulose. There will always be algae sedimentation or leaves and cellulose, but there's more of it. If it's low, it's more that the organic matter is more coming from the water column itself. So we were wondering if the organic matter source, whether it's from leaves and cellulose or from algae sedimentation, if the source and content are driving the microbial communities. To do that, we did total DNA extraction, and then the 16S ribosomal RNA PCR amplification. Ribosomal RNA found in all cells, so we can, and there's primers to look for them, sequencing, and then we looked at the sequences to determine what they are. So here we have our 24 layers and what they are. So what we see from this is that there are groups of bacteria or archaea of microbes, Bathyarchaea or Dialicocoidea. They're pretty much present throughout. But these microbes, they are actually, they thrive in sedimentary environments, especially anoxic. So it wasn't surprising to find them there. But what's interesting, and here we have my 3 zones, so group 1, group 2, group 3. What's interesting is that in group 1, which is more mangrove-like, we have the gamma proteobacteria that are mainly present in the upper part of the core, barely there in the middle, and then at the bottom, just towards the very, very end. But that's because we have this environment that is salt water that is coming, and there's a lot of gamma proteobacteria. These are actually different than those ones. Campylobacteria was mainly present in group 3, a little bit here and there in group 1, and this is probably those glitches due to because we took these sediment layers. But there's differences between the different age group, but not necessarily, it's not as obvious as we would have hoped. But we can see here, again, this is just another way to represent the same data, that the Campylobacteria, we don't see them in group 2. Gamma proteobacteria, mainly group 1, and at the bottom of group 3. So there are different groups that are different, and that's the main factor is these age groups that were determined based on the vegetation or the history of the sinkhole and the Bahamas themselves. So our organic matter source and content driving microbial communities. To do that, to answer this, we did a non-metric dimensional scaling, where you have your group 1 samples that are plotted in yellow circle, group 2, the triangle, blue triangles, and group 3, pink squares. And we did look at the composition of the microbial communities against total nitrogen, total organic carbon, and the C/N ratio, and only the C/N ratio was actually statistically valid. So that means that the origin of organic matter is important, not the amount of total nitrogen or the amount of total carbon, but really when you put the ratio and look at where is this organic matter coming from. So now what we did, we took 6 layers. So far I've told you about all 24 layers, but to do metagenomic, we were able to use only 6 layers because of the amount of material that we need. So we have 2 in group 1 in group 2, and 3 in group 3. So we did total DNA extraction, metagenomic sequencing, and we also looked at nutrient composition. So I'm showing this again because this figure is the C/N ratio, and we had a wide range. Some were very high, some were mediums, or some were very low C/N ratio. And we looked at nutrients. So the nutrients, they sort of go from high at the surface, it drops, and then in the last one, which is groundwater fed, so you have more nutrient coming from the bottom of our core too, then they go back up. So now we're going to look a little bit at the metagenomic results. So how do these samples compare? It looks like they group again based on group 1, group 2, group 3. So these 2 are more similar. This one is totally on its own, and these 3 are more similar. This isn't really informative. This is just to show that there's some sort of a gradual composition of changes in the microbes when we're so these are z-scores, and we have the microbes. So you're gonna see a couple of these, and this is based on, okay, if we take Calditrichalis, they are more abundant in the first than they are everywhere else, but that doesn't mean that they are not present in the other layers. It just means they are more abundant in the first one. But when we do metagenomic, really what we want to look at is the functional composition of the microbial communities. So we looked at the functions, and we see changes through the depth. And for example, the nitrogen metabolism is more present at the upper depth than at the lower depth. And the important one, sulfur, was also more in the upper part than in the lower part of the core. Same for phosphorus. But then when we get towards the bottom, so this sample here is very different. This one looked weird when we took it. It looked like a bunch of leaves that had been piled up on top of each other, and it looks very different no matter which angle we look at it. But you see, autotrophy was highly present. There's no photosynthesis going there, but if you're going to have preserved leaves, you're going to find these genes. And now what we're doing, and this is just preliminary data, but we are looking at these to determine who is doing what and what we have found. So we have assembled 638 species bin, which here each different line is a species bin, with 98 that are more than 50% completed and less than 5% contaminated. And now we can map the reads from our metagenomes and see where we find these microbes. And again, so here we have from 6 to 79 centimeters, so it's going up down core, and we see that there are microbes that are way more like present. And this one is not based on if it's present. If it's darker, it means it's really more prevalent at this layer. And we see this nice trend again where some are more present in the upper part of the core versus lower part of the core. And now we're going to go dig deeper and see who is doing what, so we can see the bigger picture of the ecology of Blackwood Sinkhole sediment. And our final question was, what drives microbial activity, was to look at virus-induced microbial mortality. So we did these experiments where we took sediment, made a slurry, incubated it at in situ conditions, and took samples at 0, 1, 3, 6, 12, and 24 hours. We separated the viruses and the bacteria from the sediment particles, filtered through 0.2 to count bacteria, through 0.22, it should be 0.02 to look at viruses, and then we counted using epifluorescence microscopy. So I'm going to show it to you the same way I've done. So here we have our C/N ratio, our nutrient concentration, and we are looking at viral production, the viruses and bacteria. So what we usually see when we look at these experiments is, so in light green, the light dotted green, we have viruses that will go up. They will burst the cells, so the cells will go down, and then it sort of stabilizes. This happens usually within the first 3 to 6 hours. So we did this, and based on this, we can calculate how many viruses were produced and how many bacteria died. So we have the viral production is when they're like in our bottom sample where there was a lot of nutrients, it was really low. Same for the top sample, and it seems really higher when there's fewer, when the concentrations of the nutrients was lowered. In our sample, interestingly, we have less viruses than bacteria, but this is also common in some coastal anoxic regions around the world, including the Gulf of Mexico. And based on that, we can calculate, so we've calculated viral production, we can calculate bacterial decay, how many bacteria died basically throughout our experiment, and that allows us to calculate how much nitrogen and carbon is released through the death of these cells by the lysis with viruses. And we used a software called VibCal to do that. And we basically got the opposite trend of the nutrients. So when there were fewer nutrients, we had less the dissolved organic carbon or nitrogen release. And when there's less nutrient, we get high dissolved organic DOC and DON release. So it's almost like if there's not a lot of nutrient, viruses can be there to produce nutrients, so microbial activity can continue. So with the future work with this part, so we're gonna continue our metagenomic analysis. We have some viral samples as well, so we're looking at those. Once we have analyzed our viral metagenomes, we want to computationally link viruses to their potential host to really see who is responsible for this release of carbon and nitrogen. And we want to also maybe look at these prophages, like the viruses that are in the host fractions. So in conclusions, so our first question was, are the sub-seafloor prokaryotic communities comparable to the ones inhabiting anoxic oceanic basins? Well, the microbial communities, they share some similarities, but like there was a dominance of these dihalococoidea, which are chloroflexi, proteobacteria, epsilon proteobacteria. But the nutrient source is very different because it comes from terrestrial and wetlands versus particulate organic matter that is found in the water column. So could we use sinkholes to generate preliminary data for ocean scientific drilling? Maybe if we're focusing on these similarities more than the differences, it could be used to we could leverage some of these data to better plan our expedition experiments. What drives the microbial community composition in anoxic coastal sediment? Well, I showed that the C/N ratio is a significant driver of the microbial community. That means that the source of carbon, so whether it's coming from terrestrial plants or that are surrounding the sinkhole, or from the sinkhole itself, from the algae sedimentation, this is important in shaping the microbial communities. In an environment where organic carbon is not a limiting factor, such as Blackwood Sinkhole, electron acceptors are probably the limiting factor, and that's what I've shown with the viral lysis. Maybe viruses can actually bring back these limiting electron acceptors. Could we use viral activity as a proxy for microbial activity as a whole? Maybe this is an option, something that we're looking into in the lab. And who are the main players of the microbial community? We will know more as we dig deeper into our metagenomic data. Finally, what is the extent of nutrient regeneration in subsurface sediment by viruses? Well, there wasn't necessarily a clear correlation between the viral production and nutrient concentration, but there was a correlation between the release of carbon and nitrogen by viruses that was inversely correlated to nutrient availability. Other questions that we could ask: are viruses important drivers of evolution? As we dig deeper in our metagenomic data, look for viruses, we may be able to answer this. And we may be able to answer also which members of the community are most impacted by viral lysis. I would like to thank you for inviting me, but also none of this would have been possible without my graduate student, Catherine Risley, who graduated last summer. That was her master's thesis project. And Madeline Thompson is an undergraduate research scholar. She's writing her thesis right now. She did all the viral activity experiments. And my collaborator, Pete Van Hengstum, in the micropaleontology lab, and his graduate student, Annie, who helped a lot with the C/N ratio. This work was funded by CDEBI, Texas Sea Grant, and Texas A&M as well. And if you have any questions, I will happily take them. You can also email me, and I will be happy to answer any of your questions. Thank you very much.
Jessica, there she is. Thank you very much for participating. I know when we first talked about this, we bounced back and forth between, would this be virtual, would it be live? And that pretty much took care of itself fairly early on, which actually is kind of along the lines of one of the questions that somebody emailed me. What has been the effect of the pandemic on your research? And I know you kind of hinted at that with the IODP program.
Yeah, so I had a lot of projects that were ongoing, so it didn't impact too much, but we had a lot of sequencing, that DNA that we sent for sequencing, and that took much longer than usual, and we still haven't gotten our data, and it's been months, but it's mainly because they got hit by the pandemic more than we did. But the IODP has been impacted because they had to cancel multiple cruises. So now they are trying to reschedule the cruises, but the current IODP program ends in 2023. So they are trying to reschedule the cruises and have new cruises, but they're sort of backlogged right now, trying to figure out how to overcome. But the JOIDES resolution has been running. It missed a couple expeditions, and now it's running again. Because once you're on the ship, you're basically quarantined. So if you quarantine yourself before, and if you're negative when you get on the ship, everybody should be negative.
And embedded in, there was sort of a part two to that. And I think part of that was also with the PSA, and certainly you hit on a couple of terms that have become much more broadly familiar. I think that, with the vaccine, we've heard mRNA, so you had RNA in there, viruses and different things. Any sense of just a general familiarity with the public for some of those concepts might trickle up into greater research support?
Yeah, so I'm a environmental virologist, so I don't study necessarily, like, all the viruses that I study, they don't impact, like, they don't infect humans, they infect bacteria and microorganisms that we can't see with the naked eye. But with the COVID pandemic, it has brought up some opportunities for me to at least apply for grants where we could look at the spread of COVID in like through wastewater treatment plants, for example,
Okay.
and if the treatment plants are actually working to remove the viruses or if it's going out back in the environment. Um, so it has like it brings new questions that we can start asking that are still relevant for environmental microbiology and virology.
Okay. Um, One of the things that I'm always interested in, and I think that in general, what got you interested in science in the first place?
Um, I think I've always been interested in science. I remember when I was at like I think it was in third grade, the 4-H group came and they did a couple experiments in front of us, and I was so mind blown. I was like, I want to understand the world. But so I've always wanted to be a scientist, but I didn't really know what I wanted to do. And it's only when I got to college uh, that I my so in Quebec we have community college that is before you go to university, and I really enjoyed my biology course there, and I was really interested in evolution, and they told me that if I wanted to know more about evolution, microbiology was a really good place to go because you can study the evolution of microbes. So I took their word for it and I applied for the microbiology program. And I was like, if I don't like it, I'll just switch program next year. And I'm still doing microbiology today. So.
Mhm. Um, so one of the other things is we frequently have a number of K-12 students and certainly community college students that watch these videos or watch these talks. I'm so used to the live thing versus the live stream thing. Um,
Yeah.
what would be your suggestions of potentially what some of the looking out 10, 20 years maybe, what some of the likely research job related STEM fields are, and what students should be doing now if they're in middle school, high school, or at community college in order to potentially prepare for those STEM careers.
So in geology, there's a lot of opportunities, and oftentimes, like people think geologists will not get good jobs, and I had a few slides I could have presented them, but a lot of people with careers in geology they make more money than many other people. So that's a really good opportunity. But if you are in middle school, I think just getting involved in whatever opportunity comes at us. That's the age that you're sort of allowed to not know what you really want to do in your
Mm.
future and trying things out. And I always say that like discovering what you don't like is actually a good thing, because if you try something and you don't like it, then you can check this one off your list, and then you still have other opportunities. There are many opportunities I took throughout my career that I wasn't quite sure if it was good, and I liked it, or if I didn't, I knew I was gonna just switch gear a little bit. So I think yeah, trying different things, trying to get opportunities, but sometimes even though like the opportunities are limited, so working hard towards what you like and want to do, and never believe when someone tells you you can't do it. Yeah.
Sounds like good advice. I know that you are on a sampling trek right now. We were talking before we started, sampling, is it 20 lakes in 20 days or 25 in 25
Mm-hmm.
days? So
Yeah,
the April
20,
Lake
20 and
Tour of
20.
Texas. So I appreciate you joining us this evening for the talk, and appreciate the ocean drilling program for uh, having this series and bringing top scientists from all over to be able to present on their research as well as to highlight the IODP, because it is a very important organization out there. I'd like to again thank our sponsors, DB Western, the Mill, College Foundation, the college. Thank you, Dallas, for helping make this happen and go out on the airwaves. And thank you all for attending this evening. Take care, stay safe, and hopefully we'll see you on May 11th. Bye.
Thank you.